Welcome. With this session we start the retina section, and in this first session we'll discuss anatomy of the vitreous. In the next two sessions we'll be discussing the anatomy and physiology of neurosensory retina, following which we will be discussing the basics of the retinal pigment, epithelium and the choroid in a subsequent session. The vitreous body is a transparent gel made up of collagen fibers which are separated by hydrated hyaluronic acid molecules, and there is an electrostatic attraction between negatively charged hyaluronic acid molecules and positively charged collagen molecules. The vitreous body is the largest structure in the eye, occupying 80% of the volume of the eye. 98% of the vitreous body is made up of water, and the remaining 2% consists mainly of collagen and hyaluronic acid. The vitreous body has the same refractive index as the aqueous but is twice as viscous as the aqueous. The major biochemical constituents of the vitreous body are collagen and hyaluronic acid. Collagen resists tensile forces, and 75% of collagen is type 2, which is similar to the collagen found in hyaline cartilage, and there are rare genetic disorders called ophthalmoarthropathies which affect the eye and the cartilages together. The collagen in the sclera and cornea are however different from the collagen present in the vitreous body. The collagen fibers are condensed in the vitreous cortex and are dispersed in the vitreous core, and the total amount of collagen in the vitreous is said to remain constant after adulthood. 95% of glycosaminoglycans is hyaluronic acid, and it is said to resist compressive forces and provide viscoelastic property to the vitreous body. And the vitreous also contains a high level of ascorbic acid, which is said to protect the lens against oxidative damage. Morphologically, the vitreous is divided into a central portion called the core of the vitreous, which is composed of non-branching collagen fibers arising from the vitreous base and oriented parallel to the Cloquet's canal, and a peripheral portion called the vitreous cortex. As we have mentioned, the cortical vitreous is composed of densely packed collagen fibers which are arranged in lamellae and which run parallel to the retinal surface. The anterior vitreous cortex extends anteriorly from the vitreous base, and the posterior vitreous cortex extends posteriorly from the vitreous base. The posterior vitreous cortex is around 100 to 110 microns thick, and there are two rounded discontinuities or holes in the peripheral vitreous cortex, one in front of the optic nerve, the prepapillary hole, and one in front of the macula or the premacular hole. And the anteroposterior collagen fibers have been found to extrude through these holes onto the surface of the optic disc and the macula. The circular areas shown in this diagram are called bursae, which we will be describing later in this session. Hyalocytes are cells found in the vitreous body, and they are derived from the bone marrow. They are found as a single layer of widely interspersed cells within the posterior vitreous cortex, with the highest density of hyalocytes being found in the vitreous base in the pars plana region. So the hyalocytes are primarily found in the posterior vitreous cortex. They are involved in maintaining ongoing synthesis of collagen and hyaluronic acid, phagocytosis, and immunological reactions. They obtain their oxygen by diffusion from the retinal and choroidal circulations, so that little oxygen from this source reaches the anterior segment. And the hyalocytes are important in the pathogenesis of macular pucker when they remain on the retinal surface following anomalous PVD and vitreoschisis. In anomalous PVD, there is more vitreous liquefaction than the extent of the PVD, and in vitreoschisis, there is splitting between the lamellae of the posterior vitreous cortex. The attachments of the peripheral vitreous cortex to the retina are very important, and they occur at the ora serrata, the posterior lens capsule, around the macula, around the optic nerve head, along the internal limiting membrane, and the retinal blood vessels. At the ora serrata region, the vitreous forms the vitreous base, which is firmly attached to the posterior pars plana, and the anterior retina, extending 2 mm anterior to and 3 to 4 mm posterior to the ora serrata. The collagen fibers in the vitreous base are particularly dense and most are oriented perpendicular to the surface of the retina and the pars plana, making this the firmest attachment of the vitreous to the retina, and this is very important to remember. And the posterior border of the vitreous base is the major site of retinal tears. At the posterior lens capsule, the anterior vitreous cortex is attached at the Weigert's ligament, which is an annular area of attachment of the anterior vitreous cortex to the peripheral posterior lens capsule. The Burgert space is the space between the posterior lens capsule and the anterior vitreous cortex. The patellar fossa is the depression of the anterior vitreous cortex corresponding to the posterior convexity of the lens, and the space between the peripheral lens and the vitreous base is called the annular gap. The vitreous is also attached to the internal limiting membrane of the retina and also along the retinal blood vessels. At the fovea, the posterior vitreous cortex is attached in an annular zone 3 to 4 mm in diameter, and the premacular bursa or the precortical vitreous pocket is a space anterior to the fovea devoid of vitreous. The vitreous is also adherent to the retina around the margin of the optic disc, and there is an area of Martigiani which is also devoid of vitreous and is located anterior to the optic disc. The Cloquet's canal extends from the area of Martigiani to the Burgert space and is the former site of the hyaloid artery of the embryonic primary vitreous. And this vitreoretinal attachment around the margin of the optic disc is the last to detach during posterior vitreous detachment. The aging changes of the vitreous include vitreous liquefaction and posterior vitreous detachment. With vitreous liquefaction, we find visible vitreous fibers ophthalmoscopically, where collagen fibers, no longer being separated by hyaluronic acid molecules, adhere to each other. So the proportion by volume of gel vitreous increases and that of liquid vitreous decreases with age, although the total amount of collagen in the vitreous remains constant. Vitreous liquefaction occurs first in the central vitreous, and vitreous liquefaction is known to occur earlier and be more extensive in myopes and following ocular inflammation and ocular trauma. Posterior vitreous detachment or PVD occurs following vitreous liquefaction. During PVD, liquefied vitreous exits posteriorly through the prepapillary and premacular openings in the posterior vitreous cortex and dissect between the posterior vitreous cortex and the internal limiting membrane. And detached peripapillary tissues from around the optic disc margin, mostly fibrous astroglia and collagen, may remain attached to the posterior vitreous cortex around the prepapillary opening of the posterior vitreous cortex and float as a ring in front of the retina, which is called the Weiss ring. To recap the salient points, vitreous is the largest structure of the eye, 98% of which is composed of water. The majority of the structural components consist of collagen fibers interspersed with hydrated hyaluronic acid molecules. The vitreous body consists of a peripheral vitreous cortex composed of densely packed collagen fibers and a vitreous core. Hyalurocytes are present in the vitreous cortex and are involved in phagocytosis and synthesis of collagen and hyaluronic acid. The firmest attachment of the vitreous to the retina is at the vitreous base. Other attachments include around the optic nerve head with the posterior lens capsule, in the macula with the internal limiting membrane, and along the retinal blood vessels. Thank you for listening.